Now transcription unit. So transcription unit has three regions primarily: promoter, structural gene and the terminator region. So promoter is where it marks the beginning of the transcription and RNA polymerase is going to get binded here. And structural gene is part of DNA which is actually transcribed. So this is where the transcription is going to take place. And terminator is where the transcription is going to end. Next, template strand and coding strand. The enzymes involved in transcription, which is RNA polymerase and RNA dependent RNA polymerase, is going to catalyze into one direction, which is 5' to 3'. Alli 3' to 5', illi 5' to 3'. Therefore, strand with polarity 3' to 5' will act as template strand, and strand with 5' to 3' polarity will act as coding strand, which is mesonomer, since it will not code for anything. And coding strand has sequence similar to the RNA formed after transcription, except for the change of thymine in the presence of uracil instead. So this is how the transcription process is going to take place. Now coming to gene. So the DNA sequence which will code for tRNA or rRNA molecule is defined as a gene, and cistron, which is segment of DNA, contains the genetic code for a single polypeptide, and structural genes could be of two types, which is monocistronic and mostly in eukaryotes, and polycistronic mostly in prokaryotes. And monocistronic genes have two types, which is exon, sequences which code for particular character, and they are expressed is matured in a matured and processed mRNA, and introns, they are the interrupting sequences which will not appear in the processed mRNA and matured mRNA. Next is the regulatory genes, which are the sequences which will not code for anything, but they are going to regulate the functions of these two. Now types of RNA and transcription process. So now mRNA, which is a messenger RNA. So this is going to serve as a template for protein synthesis, and DNA is transcribed to form the mRNA, which will in turn get translated to form the protein. And central dogma of molecular biology rule is going to state this. Now tRNA, it is a transfer RNA which will bring amino acids during translation, and they are going to read the genetic codes. And rRNA, which is ribosomal RNA, they work as branches of translation, and they play a structural and catalytic role at the time of translation. Now coming to transcription process. So it has three steps: initiation, elongation and termination. So firstly, initiation. RNA polymerase will bind with promoter to initiate the process of transcription, and association with initiation factor will alter the specificity of RNA polymerase, initiating the transcription respectively. Now elongation. So RNA polymerase will use nucleotide triphosphate as substrate, and polymerization is going to occur according to the complementarity. Now termination. It will occur when termination factor P will alter to the specificity of RNA polymerase to terminate the transcription process. And as the RNA polymerase will proceed to perform elongation, a short stretch of RNA is going to remain bound to the enzymes, and this enzyme will reach the termination region, the nascent. So this nascent RNA will fall off and transcription is terminated. So this is how it happens. This is the initiation, elongation and termination process. Now complexities which are associated with transcription. In prokaryotes, basically there is no clear demarcation on between cytosol and the nucleus. So therefore translation can begin even before transcription, and it can be completed also. And in prokaryotes, transcription and translation are coupled together. Whereas in eukaryotes, three different kinds of RNA polymerases are present, which is RNA polymerase 1, which will transcribe rRNA, and RNA polymerase 2, which will transcribe hnRNA, which is mRNA precursor, and RNA polymerase 3 will transcribe tRNA, snRNA and srRNA. Now, the precursor of mRNA, which is hnRNA, will contain both introns and exons. So, introns are removed and exons are joined by splicing. So, now next is capping. In this, the methyl guanosine triphosphate is added to the 5' end of the hnRNA, and then tailing. So in this, the adenylate residues are added to the 3' end of the hRNA, and when hRNA is fully processed, it is known as the mRNA, which is transported out to the nucleus to get translated. Now next is the genetic code and study of mutations. So genetic code is the one which will direct the sequence of amino acids while the synthesis of proteins is going to take place. And George Gamow proposed that 20 amino acids are to be coded by 4 bases, and then the code should be made up of 3 nucleotides, wherein 4 cube is equals to 64, which is 4 square is equals to 16, which is less than 20, so the codon was proposed to be a triplet. Now Haber-Gobin-Khorana developed a chemical method to synthesize RNA molecules with defined combinations of these bases. And now Nierenberg was the one who developed cell-free systems for protein synthesis, which helped to code the deciphered. Now the enzyme known as Severo-Okai enzyme, which is the polynucleotide phosphorylase, was the one which helped to polymerize the RNA with defined sequences in a template independent manner, and it finally gave rise to the checkerboard of genetic codes. So this is the checkerboard. Okay. Now salient features of genetic codings were: codon is a triplet wherein 4 cube is 64 and 61 are going to code for amino acids, while 3 are the stop codons, and 1 will code for the single specific amino acid, and codons are the unambiguous ones, and codons will degenerate since amino acids are coded by more than one codon respectively, and genetic code is universal. One codon is going to code for same amino acids in all the species, and codons are read in a continuous manner. So there is no breakage. If you are going to read AUG, then you are going to read GCC and ATG, all these continuously. Okay. Now next, AUG has dual functions wherein it codes for methionine, and it is also going to act as a start codon. Now effects of mutation on genetic codings. Mutations will include insertion, deletion and rearrangements, and mutation will result in change phenotype and diseases like sickle cell anemia, which is change in GLU-val in genetic coding for beta globulin chain of this hemoglobin, and such mutations are called the joint mutations. And insertion or deletion of single base pair will disturb the entire reading of the frame in mRNA, and such mutations are called the frame shift mutations. Now frame shift mutations will hold the proof of the fact that codon is a triplet, because if we insert 3 or multiple of 3 bases followed by the deletion of same number of bases, then reading frame will remain unaltered. Structure of DNA and process of translation, regulation and gene expression. So tRNA, it is an adapter molecule, and on one hand it is going to read the genetic code, and on the other hand it is going to bind the specific amino acids. And tRNA has an anticodon loop where the bases are complementary to mRNA code and amino acid acceptor end, where it binds to the corresponding amino acid. Now initiation tRNA. So this tRNA is essential for initiation of translation and has a AUG in anticodon loop and MET in the amino acid acceptor end, and there is no tRNAs for stop codons. So this is the structure of tRNA molecule. And then is the translation, wherein the mRNA will contain the genetic information which is translated into amino acid sequences with the help of the tRNA. And amino acids are polymerized to form polypeptides, and these amino acids are joined by peptide bonds. Now first of all, charging of tRNA, which is the amino acylation of tRNA is going to take place, and in this amino acids are activated in the presence of ATP, and then they are linked to their corresponding tRNAs, and ribosomes are the work branches for translation here, and these have two subunits, which is large subunit and small subunit. So now the smaller subunit will come in contact with the mRNA to initiate the process of translation. Whereas translation unit is an mRNA is the region flanked by start codon and stop codon, and untranslated regions like the UTRs are the regions on mRNA which are not themselves translated but are required for efficient translation process, and then they may be present before the start codon, wherein 5' UTRs, or after the stop codon, which is 3' UTR. An initiated tRNA recognizes the start codon, which is initiation, and then tRNA amino acid complexes will bind to their corresponding codons on the mRNA, and base pairing is going to occur between codon and mRNA and tRNA anticodon. Now tRNA will move from codon to codon onto the mRNA, and the amino acids are added one by one, elongation. Now release factor will bind the stop codon to terminate the translation, termination. Now regulation of gene expression. So regulation of gene expression could be exerted at the following levels. One, the transcriptional level following a primary transcript, and processing level, which is splicing, transport of mRNA from the nucleus to cytoplasm, and translation level. In addition, metabolic, physiological and environmental conditions regulate the expression of genes, and expression of genes coding for enzymes is required only when substrate for the enzymes is available. For example, lactose, which is beta galactosidase, will give rise to glucose plus galactose, and E. coli synthesizes beta galactosidase only when lactose is available. Now regulation in prokaryotes. So gene expression is regulated by controlling the rate of transcriptional initiation, and the activity of RNA polymerase at a given promoter is regulated by accessory proteins, and the accessory proteins will affect the ability of the promoter to recognize the start sites. And now a regulatory protein could be activator or a repressor, and accessibility of the promoter is also affected by the operators here. And now operators is a region located at adjacent to promoters, and each operon has a specific operator and a specific repressor, and usually operator will bind to the repressor protein. Now regulation of the Lake operon. So operon is the arrangement where polycystonic gene is regulated by a common promoter and regulatory genes. And Lake operon, TRP operon, His operon, Val operon are the examples of such systems. Now the elucidation of Lake operon as a transcriptionally active system was first done by geneticist Jacob, who was a biochemist Monod. And genes consisting of Lake operon were the inhibitor genes, which was the I gene, and it coded for the repressor of the Lake operon, and then Z gene, which was structural gene, and it coded for the beta galactosidase, which is lactose galactose plus glucose, and Y gene, which was also structural and coded for permease, which increased the permeability to the cell, of the cell to beta galactosidase, and alpha gene, which was also structural, and this coded for transacetylase. And all genes involved in Lake operon are required for metabolism of the lactose. And inducer, lactose acts as inducer for Lake operon, since it will regulate the switching on and off of the Lake operon. And if lactose is provided to the growth media of bacteria in the absence of any other carbon source, so now this will act as in the absence of any other carbon source, and then it is transported inside the cells by permeases. And for permease to be present and lactose to enter inside the cells, low level of expression of Lake operon must be present all the time. Now regulation of in the absence of inducer. So in absence of inducers, I gene will transcript to synthesize repressor mRNA, which will translate to form the repressor. And this repressor is going to bind with the operator region of the operon, and it is going to prevent the RNA polymerase to transcript genes like Z, Y and A, which are negative regulations here. And therefore in absence of products of these genes, metabolism of lactose is going to cease. So this is the absence of inducers. Now regulation in the presence of inducers. So inducers will bind to the protein product gene I, which is the repressor, and it is going to inactivate it. And this inactivated repressor is unable to inactivate the RNA polymerase enzyme, and therefore Z, Y and alpha genes will synthesize their respective mRNA, and in turn they get translated to form the beta galactosidase, permease and the transacetylase respectively. So this is in the presence of all the enzymes, the metabolism of lactose is going to proceed in a normal manner. And HGP, Human Genome Project. So joint venture of US Department of Energy and National Institute of Health, which is NIH, later joined by Wellcome Trust, which is from UK. So launched in 1990 and it was completed in 2003. And this project worked towards the determination of complete DNA sequences of the humans. Now DNA is the storehouse of genetic information, and it is a determining sequence of base pairs and can also solve medical, agricultural, environmental and evolutionary mysteries. So medical is wherein we have got so many genetic disorders, and agriculturals by producing these hybrids by changing their DNAs, and environmental and evolutionary changes also relate those. Now relationship of HGP with bioinformatics. So mathematics and HGP in concluded together. So human genome, which is genome, refers to the total genes of present in the human beings, which were 3 into 10 to the power 9 base pairs. So now cost of sequencing one base pair is $3, and cost of sequencing 3 into 10 to the power 9 base pairs is $9 billion. So enormous sequence data so generated would be required 3,300 books containing 1,000 pages each for a human genome. Therefore, for storing, retrieving and analyzing these enormous data, a new branch of biology has been developed, which is called as the bioinformatics. So genomes have many non-human models like bacteria, yeast and the Coanohabitates elegans and Drosophila. Plants which are rice and Arabidopsis have also been sequenced with the help of this bioinformatics. Now methods to identify the genes are two methods. One, the identifying EST, which is express sequence tags, and sequence annotation. So now ESTs, as the name suggests, this will refer to the part of DNA which is expressed, that is transcribed as mRNA and translated into proteins. Thereafter, it basically focuses on the sequencing part of the denoting a gene. And then annotation. So in this approach, entire genome, which is a coding and non-coding, is sequenced and then function it as associated region in the genome. Now genome sequencing. So DNA from the cells is isolated and randomly broken into fragments of smaller sizes, and these fragments are cloned into suitable hosts using the vectors, and cloned fragments are going to amplify in the host. Amplification will facilitate an easy sequencing, and common vectors used are the BACs, which are bacterial artificial chromosomes, and yeast artificial chromosomes, YACs. Now common hosts are bacteria and yeast. Automated sequences are used to sequence the smaller fragments, which is the Sanger sequencing, and then the sequences so obtained are arranged based on the overlapping regions with them, which is the alignment. And alignment of sequences is also done automatically by computer programs, and then these sequences are annotated and assigned to each chromosomes. Now preparation of genetic physical maps and genomes. Now two methods are used, which is restriction polymorphism and microsatellites. So restriction polymorphism is a specialized enzyme called restriction endonucleases, which is the REN, are used to cut the genome in a specialized site called the restriction endonuclease recognition site, and maps are prepared based on it. And microsatellites are the repetitive DNA sequences. Now observations of HGP. HGP human genome consists of 3 into 10 to the power minus 9, which is 164.7 million nucleotide bases, and on an average gene consists of 3000 bases, and however the size of gene will vary ultimately, and largest gene is the dystrophin, which is 2.4 meter bases, and total number of genes in human genome is 30,000, and over 50% of discovered genes have unknown functions, and less than 2% of genome is coding, and large portion of genome will constitute of repeating sequences. Now repetitive sequences have no coding function here, and they were repeated over 100 to 1000 times, and they may have role in evolution, chromosomal structures and dynamics. And chromosomes with most genes was chromosome 1, which was 2968, and chromosome with fewest genes was chromosome Y, which is 231, and SNPs, which is the single nucleotide polymorphism, occurred about 1.4 million locations in human DNA, and they were believed that these have the significance of explaining diseases and evolutionary history of the human beings. Now DNA fingerprinting introduction. This is very important. So DNA fingerprinting is a method of comparing the DNA sequences of any two individuals, and 99.9% of base sequences in all human beings were identical, but the 0.1% was the one which made every individual unique. So for example, if you take your hand, not everyone's thumb will be of the same fingerprint. So it can be recognized very differently. Though you have the similar structure of your thumb will differ from one another. It is really difficult to the time consuming task to sequence and compare all 3 into 10 to the power 9 bases in 2 individuals. So instead of considering the entire genome, certain specific regions called the repetitive DNA sequences are used for this DNA fingerprinting comparative study. Now basis of this DNA fingerprinting was DNA repetitive DNA, and this was separated from bulk DNA, and since it appeared distant peak during density gradient centrifugation, and major peak was formed by bulk DNA and smaller peak was from satellite DNA. So satellites are of two types, microsatellites and mini satellites. So depending upon the base composition, length of the segment and the number of repetitive units, it was divided as micro and mini satellites. And satellites were not coding for proteins but have a major role to play in DNA fingerprinting. So polymorphism is actually a result of mutation, and a germ cell mutation which can pass to the next generation through sexual reproduction will give rise to polymorphisms in populations. And in other words, an inheritable mutation is observed in higher sequences in a population. This is known as polymorphism. Now polymorphism arises normally in non-coding sequences because mutations in non-coding sequences will not affect an individual's reproductive ability. Now coming to the methodology. So it is VNTR, which is variable number of tandem repeats, are satellite DNAs which show high degree of polymorphism, and VNTRs are used as probes in DNA fingerprinting. And first of all, DNA from an individual is isolated and then cut with the help of these RENs. Now fragments are separated according to the size and molecular weight on gel electrophoresis. So there are various gel electrophoresis tanks which are used, and fragments separated on electrophoresis gel are blotted on a synthetic membrane such as a nylon or nitrocellulose membrane. So there will be the fingerprints like this. Okay, you will have bands which would have created on this blotting paper. So immobilized fingerprints are hybridized with VNTR probe, and hybridized DNA fragments can be detected by autoradiography. And then VNTRs will vary in size from 0.1 to 20 kb. Hence in autoradiogram, band of different sizes will be obtained, and these bands are characteristics for every individual, and they are identical in every individual except identical twins. So they are different for every individual except in identical twins. So this is how the electrophoresis is done, and these are the bands. Now applications. So it is widely used for forensics since every DNA of every tissue from an individual has the same degree of polymorphism. And DNA fingerprints forms the paternity testing since child will inherit polymorphism from both of its parents. And then it can be used for studying genetic diversity in population and evolution respectively. Thank you.